So, yes. It said I'm voting for tacos. I needed to find something we could all agree on because I feel so divided. But can't we all agree on tacos, guys?
I want a good taco after class.
Oh my God, tacos are like my favorite food in the history of
Can we have
foods.
tacos like the last day of this class?
Are you going to bring tacos? Then yes, we can have them. Because I have some crap to do. I don't know if you know that, but I'm from Texas. I like in mourning for Mexican food every day of my life. Like you come to Utah and I'm like, where is all the food?
Sushi.
What? Oh, chili berry is so good. All right, focus. It's Friday for all of us. We're having trouble. Okay, let's get together. Okay, so things you want to know. First off, quiz on Monday. It's going to be over the heart. Okay? Second, test on Wednesday. Here's what it's going to cover. Your test is going to cover brain, which is probably the most challenging part. Spinal cord and autonomic, which is the next set of notes, right? And heart. It is not going to cover vessels. I'm going to roll vessels over to the fourth test because the brain got rolled over to the third, and it seems like that's okay. That's good enough, right? In addition, this will allow you to have vessels in lab before you're tested on it, right? So I'm awesome, don't you think? I just decided that when I said it out loud. I'm like, I'm so cool. So brain, spinal cord, autonomic, right? So 3 sets of notes. But the brain was kind of long, right? It's a pretty long set of notes, and it's kind of hard to get it together. So tell us again, where are they? Cerebrum. Yeah, see? You got We will never forget. Okay, so, and then something else I wanted to tell you. Oh, next, I just want to fart around today, basically, but I'm going to get something done. So next week, who has lab on Thursday? Anybody? Next week, yeah. Not your lab, but labs exist on Thursday. Okay, so none of you guys have lab on Thursday, generally? Okay, that's great. That's awesome, because I'm going to tell you something that is really useful for the rest of your academic career. Always take labs in the middle of the week, like Wednesday if you can, because you always end up missing Thursdays and Fridays or Mondays, right? And they never lop off any of the material. You just like have to learn it more quickly. Because the reason I'm saying that is because next week in lab, they do all of the vessels, respiratory system, and digestive system. Because they're going to miss Thanksgiving, right? So, I know, that's what I said. So all you guys are smart enough not to take Thursday labs. Don't take Monday labs ever either. No, you don't get Wednesday off. Glad I could help in retrospect, right? You'll know for the future. Okay, help me focus. This is a team effort. Help me focus. I want to go home and have some tacos too. So, okay, so we are going to talk about blood vessels today. So I want to tell you, first off, that you got a whole bunch of them. You got 60,000 miles of blood vessels in an average sized person. Any of you guys get to see the body donor, bodies or body worlds? Okay, if you didn't, please see it the next time. It's so freaking awesome. One of the things they do, in addition to the plastinated specimens, is they do a preparation in which they impregnate all of the vessels, including the capillaries, with latex, and then they dissolve off all of the other tissues so that you end up with like a basic map of the cardiovascular. I know, isn't it cool? And you can see like where there's more vessels, you can imagine that there's a lot of need for blood flow. So look at the head and then look at the kidneys. Do you see that? The kidneys, like their whole outline is right there. So you can see sort of the blood flow needs. And this one is just the head, which I think is even cooler. Oh my God, it's so cool. Isn't that amazing? Why, he is a Spider-Man villain, yeah. Well, it's just the colors that make you think of Spider-Man. Okay, so you've got about 60,000 in an average sized person, about 60,000 miles. Most of those are capillaries, of course, because capillaries are incredibly branched. There's trillions and trillions of capillaries. So let's start talking about the basic structure of a blood vessel, and then we'll get into some of the specifics later. So the basic structure of a blood vessel, we'll start with capillaries. They're the easiest thing to imagine the structure of. So let's look at a capillary structure right down here at the bottom. So the way that a capillary is structured is for maximum gas exchange, they need to be as thin as possible, correct? Isn't that the place that gas exchange occurs? So you need to be highly branched, which isn't shown in this picture, but capillaries are highly branched. And then the thicker the wall gets, the more of an impediment to transport it becomes, right? That makes sense? So it's as thin as possible. It's one layer of simple squamous epithelium. It's got a basement membrane underneath, but there is no connective tissue, right? So you get ready exchange in and out of capillary beds for gas exchange. So that's the simplest structure that you have. And then also after that, we will compare an artery to a vein. And instead of memorizing the difference, I want you to think about why they are different. Why is an artery different than a vein? Okay, so first off, all vessels except capillaries have these three layers, and they are called tunics, like a coat or a tunic, right? Tunica intima or interna, those two are both used. Tunica media, which is the middle coat, and tunica externa. Good? Okay, so what are they composed of? And how are they different from an artery to a vein? The tunica intima on both of them is nice, slippery, simple squamous epithelium. Remind me why it's very important that the inside of your cardiovascular system is slippery. Keep it going so that you don't clot inappropriately, right? Okay, so that's the tunica intima. We'll come to the valves in just a second. They're both super slippery. And then if you move deep to the epithelial tissue, you're going to run into a little bit of connective tissue in both of them, which is not surprising at all. And then in an artery, you have a layer of elastic that you don't have in a vein. See if you can conceptualize. I know you might not have actually thought about this before or have been taught it before, but can anybody conceptualize and verbalize why elasticity is more important in an artery than it is in a vein? Yeah, and not just pressure, but also variable pressure. So I want you to think about how arteries receive blood. They receive it in squirts, don't they? A big squirt of blood as a ventricle contracts, and then in between squirts, the pressure will go back down, right? Big squirt of blood, and the pressure goes back down, right? Does that make sense? So what they need to do is they need to have elasticity in their wall so that they can balloon out a little bit and snap back, right? We don't want them to balloon out and stay out. That's an aneurysm, right? We want them to balloon out and snap back. Does that make sense? So the elasticity that you have in the wall of an artery is not as necessary in a wall of a vein, because if you think about it, how does a vein receive blood? In trickles from the other side of a capillary bed, right? By the way, you don't even have a pulse in veins. You have a pulse in your arteries, right? Your arteries go boom, 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 and you can put a sphygmomanometer on, and you can assess your systemic arterial blood pressure. And if you looked at a vein, there's almost no detectable change in a venous blood pressure, right? It's just pretty much constant venous blood pressure. That makes sense to you guys? Okay? All right, so this one doesn't need a layer of elastic right there, is what I'm telling you. There's like not an extra layer of elastic. And then what we're going to do is we're going to get to this layer. The tunica media is composed primarily of smooth muscle. So now, conceptually, same kind of question. Why do I need more smooth muscle in the wall of an artery than I do a vein? Any idea? Go ahead. Yeah, they both do vasoconstriction and vasodilation, but the artery, do you remember how the skeletal muscle around your joints is not always just about wiggling them, it's also about stabilizing them, right? So if I have thick musculature in the wall here, it's because it allows me to not balloon out too far, right? If I just keep muscle tone in the wall of the artery, then when it gets a squirt, right, it doesn't balloon out so far that I have an aneurysm, right? It's like holding on to the structural stability. And then over and above that, we're also going to talk about constriction and dilation, right? So, but can you see why there's a difference in the wall, in the thickness of the wall right there? So they've both got smooth muscle, of course, in the tunica media, but this has a much thicker smooth muscle, okay? And then it has another layer of elastic that you don't have here, right? It's got an internal elastic membrane and then an external elastic membrane, okay? So again, you don't need as much elasticity, and then both of them have a tunica externa, and the tunica externa is primarily composed of connective tissue. Now I know you would want to think that they're just like hanging out and they would be composed with, like, covered with epithelial tissue, but when you start looking at vessels next week, let me tell you that if you, for instance, dissected an arm and then opened it up and looked for the blood vessels, you're not going to see them just running along the surface, because blood vessels are woven into the tissues around them. That's why they have connective tissue on the outside. So why do you see the blood vessels when you look at the arm? It's because we actually slid a probe underneath them and isolated them so that you could see them, because it's much harder to find them if they're still woven into the fat and connective tissue and skeletal muscles. So they're woven into the tissue. So the outer layer of them is made of connective tissue, not epithelial tissue. That makes sense? Okay, so those are your 3 tunics. Oh, wow, I didn't even know I could do that. Cool. I was like, woo, magic. Okay, oh, let me fix it. It's bothering me now. Okay. Okay, so now let's look at this. So if you look at this, and I want to show you that, okay, so this is a tube, right? And this is how the smooth muscle is arranged around it. So now I want to ask you, if I contracted that muscle, would it cause constriction or dilation? Increase or decrease the size of the tube? Decrease, which is constriction or dilation? Constriction, exactly. So if you stimulate the smooth muscle, it will cause constriction in the vessel, right? Okay, now, just to put this together with stuff that we've done before, controlling whether that occurs is stimulated by the autonomic nervous system, because this is what kind of tissue? Smooth muscle, exactly. Does that make sense? Right? Putting it all together again, making it into one story, right? So autonomic nervous system will determine primarily whether those will constrict or dilate, okay? All right, so now let's talk about arteries in a big picture. So arteries are vessels that carry blood from the heart to the tissues. They're also called efferent vessels, which should make total sense to you. And remind yourself, I know that you know this, arteries carry blood in the what circuit? Sorry, arteries carry oxygenated blood in the what circuit? Systemic, and they carry deoxygenated blood in the pulmonary. Good. Okay, so the vessel wall is thicker and stronger in an artery than it is in the vein, and which is important because the pressure is always greater. Because of the structure in the wall of an artery versus a vein, arteries have 2 primary characteristics: elasticity and contractility. So now, without looking at the labels, can you tell me here which of these is an artery and which is a vein? Bottom is an artery and top is a vein. Now, I know it's counterintuitive, but it is true. Veins also have a larger lumen than arteries, which will actually be important because a smaller lumen will maintain the pressure and therefore the speed will be higher, right? And an artery is like on its way to work, right? And a vein is on its way home, right? It might as well fart around a little bit, because as soon as you get home, somebody's going to make you do something and you know it, right? With me, they're going to want dinner, right? They're like, somebody's always hungry. This is one of the most disappointing things about being a parent I ever learned. They are constantly hungry. Like, you literally just fed them. Like, I am constantly trying to keep up with people who are hungry. I'm like, Jesus, I just made like a 5-course meal, and then like, can I have a snack? I'm like, no, go away. Or you make a 5-course meal and they're like, can I have cereal? Shut up. Okay, so, here's one little interesting thing. We'll talk about it a little bit later. I forgot to talk about it in the last class, but it's okay. I'll get it later. Okay, so one of the other things is that arteries sometimes, so let's say I've got a really precious capillary bed. So this is a capillary bed, okay? And what I have sometimes in really precious capillary beds, like my brain is a pretty damn precious capillary bed, right? What I have there is I have alternate routes of circulation, because if I had an artery going into this capillary bed, right, and heaven forbid something happened to the artery, then I'm freaking dead, right? Totally dead, right? So what often happens is you have this redundant circulation flow to important capillary beds, in which you've got at least 2 arteries going in, right? So it's redundant, but in addition, you've also got this between them. This is called an anastomosis. What it does is it allows you to reroute blood flow patterns if something occurs. Because if I had blood flow going into the capillary bed here and blood flow trying to go in here, then what I can do is I can actually reroute through there as well. Does that make sense? So this is really important with your brain. Your brain is a phenomenal example of an anastomosis. You have 4 blood vessels going to your brain, 4 big arteries going to your brain, and you're not going to die if one of them gets blocked, and that's actually what happened in my brain. I'll tell you guys about it next time. So if you can't get blood up that way, it'll go around to get blood to the same capillary beds. And you can see how that was like a really phenomenal evolutionary adaptation, right, to actually develop collateral circulation patterns. So the big picture is this is called collateral circulation, like lateral. There's two of them, and they're collateral, like redundant circulation patterns. And then the alternate route between them is specifically called an anastomosis. Okay, so now let's move to kinds of arteries. So there are actually three different categories of arteries. As you move away from the heart, the big picture is that elasticity is most important closest to the heart, yeah? And then arterioles are going to be the most active in vasoconstriction and vasodilation because they'll control blood flow patterns to capillary beds. So if you start at the heart, and this is, by the way, systemic circulation that it's showing here. For all practical purposes, unless I tell you otherwise, we're going to be talking about systemic circulation, right? Pulmonary circulation will be a small part, important, but small part of the story that we're talking about here. So the vessels that are closest to the heart are going to be elastic arteries. They're going to receive the highest pressure, and elasticity is going to be super important in those. So name me two vessels that you know without me telling you that are elastic arteries. Aorta and there's two arteries, only two arteries attached to the heart. Pulmonary trunk. So the aorta is the elastic artery in the systemic circulation. The pulmonary trunk is the elastic artery in pulmonary circulation. Okay, so we're going to start with the aorta. The aorta is a classic example of an elastic artery. So even though it has a whole bunch of muscle in its wall, its primary job is elasticity. It's got a really wide lumen, and it receives big squirts of blood, so it needs to basically sort of balloon out, snap back, balloon out, snap back. Only balloons out a little bit, but it does balloon out. See if you guys can conceptualize this. It's always a little hard to figure out whether it's clear why this is true. What if I told you that the aorta almost never constricts? So you've got tone in the wall because of the smooth muscle, but all of that smooth muscle almost never constricts. Can anybody explain why you would not want your aorta to constrict? Why?
Because you want more blood. You don't want to make less blood go through, so the blood will like expand.
So it's going to go out, and what would happen to where would it get trapped if I constricted my aorta? It gets, right? Do you see that, like, we need it to be like structurally stable in the wall, but we really don't need it to get any narrower, right? So you really need the blood to keep going. Now, there are disorders in which your aorta constricts, and just so you know, like reduces your blood flow to your brain and your heart and can cause a heart attack or a stroke. It's definitely going to make you lightheaded. You can imagine that, right? Okay, so even though all of that muscle in the wall of an elastic artery, it's far more active in elasticity than it is in contractility. That makes sense? And then as you move further away, the elasticity becomes less important as you move further away, and the muscular action becomes more important, right? So you get a little more likely to be active in constriction and dilation. So the muscular arteries are the next category of arteries, and those are most of the arteries you're going to meet next week in lab. So for instance, you got a brachial artery, right? That's going to be a muscular artery. You got a femoral artery. You got an anterior tibial and a posterior tibial artery. The vast majority of the named arteries you're going to learn next week will be muscular arteries. They've got both muscularity and contractility, muscularity and elastic fibers, but they become a little less important that they're elastic and a little bit more important that they're muscular. And then by the time you get down to the smallest arteries, which are called arterioles, those don't have individual names, thank goodness. They're too variable between people, and there's too damn many of them. There's like 10,000, 15,000 arterioles. So, but these guys, elasticity is less important than it was before, but they're really wiggly. These are the wiggliest arteries that you have because they are controlling blood flow patterns to capillary beds. Okay, so let's go back to the same flippin' example we've used 3,000 times this semester, and let's talk about when the tiger walks into the room, how does it change the arterial flow patterns, okay? So tiger walks into the room. I had a bunch of blood going to my digestive tract because I was totally cool, and those vessels, the arterioles leading to my digestive capillaries, were dilated, right? And now a tiger walks in, okay? And what happens to those capillaries? Constrict. what happens to the capillaries that are leading to my skeletal muscles? Dilate. So what arterioles do is they control this sort of disproportionate flow pattern through capillary beds, because you don't always need the same amount of blood in all places, right? And it can change based on the body's needs. And the arterioles are the gatekeepers to the capillary beds. And so they're super, super wiggly, and they're wiggly in ways that like change based on what you're doing or not doing. Is that good? Okay, so that's what arterioles do. Okay, so arterioles. Capillaries are next. So capillaries, basically capillaries are teeny, teeny, tiny little guys. The smallest capillaries that you have are only as wide as about a red blood cell. So they have to even line up to go through those capillaries. So they're really, really tiny, the smallest ones. And then they're really, really thin. And like I said, their primary function is transport, right? They allow gases in and out, but not just gases, also nutrients and waste products. Everything can move in and out through capillaries. And you have more capillaries, like we said before, in areas that are more active, right? So you can see that there ain't a whole hell of a lot going on there compared to your like kidneys, right? So, and there's only a few places in the body where you don't have a capillary within millimeters of the cells. And those places tend to be relatively inactive, and also they have trouble healing if you, unless they're super thin. So of course, remember, you don't have any capillaries in like epithelial tissues and the epidermis, but that's okay because it's microscopically thin, right? And it can get its nutrients by diffusion. You don't have any capillaries to speak of in hyaline cartilage. And so if that gets damaged, it has trouble healing. You have very few capillaries in tendons and ligaments, so it's good not to damage those either. And then you don't have any in your cornea or the lens of the eye. Luckily, those don't get damaged very frequently. But you know, if you damage your cornea, you're kind of hosed. A lot of times you'll have to have a corneal transplant. So there's most places in the body have a lot of capillaries. Okay, so when you look at a capillary, and this is, of course, a systemic capillary that we're looking at here, we have an arterial end and a venous end of a capillary. And if it is a systemic capillary, then the arterial end will have what? High or low oxygen? High oxygen on the arterial end, and on the venous end it will be low, right? If you were going through a pulmonary capillary bed, the proportions would be exactly opposite, right? So what we are seeing here is, of course, gas exchange as you go through capillary beds, and that should be good. Okay, so the arterial end is also going to be higher pressure than the venous end. And so I want to talk about that. So why does the pressure change as you move through a capillary bed? There's a few reasons, and some of them you can come up with just thinking about plumbing. So why would the pressure on the arterial end of a capillary bed be higher than the pressure on the venous end of a capillary bed? Well, yeah, it's coming from one space, right, and then splitting. So if you came from 10,000 arterioles and then split into 100 trillion capillaries, right, that's going to drop the pressure. Do you see? But there's a couple other plumbing reasons. What are those?
It's closer to the pump.
Yeah, it's closer to the pump at that end, right, and then it's getting further away from the pump. That makes sense? And there's one more reason, which you may or may not know. Capillaries are at least partially porous, which means you lose fluid as you go through there. Does that make sense? So on the venous end of a capillary bed, we are further away from the pump. We've gone from 10,000 to trillions, right? And we've lost some of the fluid on the way through. So they are distinctly lower pressure on the venous end than they were at the arterial end. Does that make sense? Okay, so now let's look at the structure of a capillary. We already know basically that they are thin-walled. And then let me tell you one weird thing about capillaries that might kind of surprise you. Okay, so we're going through this capillary. So I want you to imagine for a second that this capillary bed right here is in the wall of a uterus, okay? And now I want you to imagine that a tiger just came in, right? And I need that blood out of that capillary bed, right? I don't need it to fart around with the uterus. We don't have time for that stuff. So what's going to happen is capillaries quite often have these things. I know that there's not any smooth muscle in the wall of a capillary, but stick with me for a second. Here's the arteriole, okay? And they just took off the tunica externa so you could see the muscle, okay? And here's a venule, okay, and they're not worried about the muscle on that side. And so this is before the tiger came in. It's just farting around on its way through the capillary bed, right? And then a tiger walks in. Well, what happens is you have these pre-capillary sphincters that can shut down all of the blood flow through the sides to make sure that it goes quickly through the capillary beds and gets back into circulation. Do you see what that does? It forces it through a thoroughfare channel when you don't have time to wait for it to go through the capillary bed and you need it elsewhere. Go ahead.
Is that one of the reasons why it's like you can lose a baby when you have traumatic experiences?
Oh, that's an interesting question. That's probably got to do with part of it. It would change blood flow to the wall of the uterus, but it's probably also hormonal. You've got stress hormones that are released during that time. Go ahead.
So the gas exchange then does not occur with someone?
It doesn't occur as frequently, right? So what you would do is immediately cut off the metabolic activity of that organ back down to baseline levels instead of like high levels, right? That makes sense? Simultaneously, though, remember that while you were doing this, you would also be inhibiting the smooth muscle in the wall of the uterus, so it's okay to do this, right? So you're saying not as much blood flow through, right? But you're also saying slow the hell down, we got other things to do, right? So the messages are not contradicting one another, right? With sympathetic stimulation, you would say, you need to calm down, smooth muscle in the wall of the uterus, and then you say, okay, you're calming down and you also don't need as much blood. Is that okay? So you never cut it off so much that the tissue dies, right? You just cut it down to baseline level. Go ahead.
Is that kind of like, does this relate to like anaerobic exercise, like sprinting? How after you finish sprinting, your muscles are screaming because they don't have any oxygen?
Not directly, because sympathetic stimulation actually increases blood flow to muscles. With anaerobic training, what happens is you're using your muscles more quickly than your cardiovascular and respiratory system can catch up. So it's vaguely related, but it's more metabolic than it is like this whole story, because you actually are trying to send as much blood flow to muscles when you're exercising as possible. So sort of, like asterisk, yes, sort of. Okay, so you guys kind of understand the precapillary sphincter idea? Okay, so now let's look at the types of capillaries, because not all capillaries are the same. So they're all thin, right? They all have a basement membrane at most, but they're not all the same. So if you look at these capillaries, you can see that as you move from this side, the left side, to the right side, they're becoming more porous, yes? More porous, more holy, not, but the other kind. So more porous, okay? And in addition, can you see that if you become more porous, you become less protective, right? So your capacity for transport goes up if you go this way, right? But your capacity for protection goes up if you go that way, right? So basically we have different needs for capillaries in different parts of our body. This one right here is called a continuous capillary, and a continuous capillary has tight junctions between all of the cells. And in order to get from the capillary into the tissue, you're probably going to have to go through a cell, not between a cell. So can you think about any place in the body that you're pretty darn sure that you have continuous capillaries? What? Superficial areas, you do have them in the dermis, yeah. Where else? Brain, exactly. Yeah, the blood-brain barrier has to have these kind of capillaries, right? As a matter of fact, the blood-brain barrier is like continuous capillaries plus. They're called continuous complete capillaries. Like, I am not messing around capillaries, right? So those are the tightest of tight capillaries in the blood-brain barrier, right? And then we're going to move up to these, and these are called fenestrated capillaries. And fenestra means window or opening. So what's going to happen with these is you have fewer tight junctions. In addition to the fewer tight junctions, if you took a simple squamous cell, you could actually sort of flatten it out. It looks like a fried egg. And then what you have in areas, those little things that look like pores, do you see them? They're not actually holes. What they are is actually double-paned windows. So what happens in those regions is that you kind of squish out all of the organelles and most of the cytoplasm so that you got a really thin area, so it's easier for transport to go in and out, right? And you have a bunch of those through the cells, and it will increase the transport rate. That makes sense? But it's not giving up completely on protecting, right? It's still allowing for some protection, okay? So where do you have these? You have these where you need transport with control. For instance, lining the digestive tract, just behind the digestive tract, think the small intestine where you do most of your absorption, right? So you need to absorb a tremendous quantity of nutrients. But there's a bunch of funky stuff in the things that you eat. I know we don't like to think about it, but none of our food is sterile, and some of it has toxins and pathogens in it. Do we really want to just absorb everything willy-nilly? No, we want to have some level of control over the things that we absorb, right? So we want high levels of absorption with control. Or if you're in the kidney, you want to push some stuff into the urine, but do you want to hold on to, for instance, your blood cells and your clotting proteins? Yeah, so you want transport with control there. So that's called a fenestrated capillary. And then the last type of capillary is the one that is the wide open. It even has an incomplete basement membrane, has hardly any tight junctions to speak of, big spaces between the cells. Where would you want to use that one? You're going to use that one where you need to be moving whole cells in and out. So for instance, I don't know if you guys have learned about the liver function yet, but you're going to learn from me next Friday. But the liver is pretty damn interesting. One of the things that happens is all of the blood that you absorb from your digestive tract, okay, which is not sterile, just so you know, there's all kinds of funky stuff that will get through, even with the fenestrated capillaries. What I need to do to that blood is I need to send it through the liver before I send it back into circulation. So that the liver can kind of like oversee what's going to go into circulation, can make sure you're not sending a bunch of poisons and toxins and pathogens into circulation. So you send it through the liver. The capillaries that are in the liver are these big, wide sinusoid capillaries. So they're wide and they're curvy. What will that do to the speed of blood flow if you're wide and curvy? Slow it down, so that's going to be important. In addition, they've got big holes on the side. So here's the blood that's going through this capillary. And what happens is the big wide sinusoid capillary has on top of it macrophages. And the macrophages are looking at the blood as it goes through, and as something funky goes in, like, oh, that's an E. coli. It's like, nope, not on my watch. It scoops it out of there. Does that make sense? So you need to be able to bring whole cells in and out, right? That make sense? Okay, so that's a sinusoid capillary in one location. Now I'm going to tell you the other location, and I want you to help me figure out why you have sinusoid capillaries in that. The other classic location for sinusoid capillaries is in the red bone marrow. So can anybody hypothesize why you would need sinusoid capillaries in red bone marrow? What? Yeah, so where blood is made. So now what are we going to do? We're going to put it in. Exactly. We need big, wide openings because I have to put whole blood cells, right, from the red bone marrow into the bloodstream, right? So that's your two classic examples of sinusoid capillary places. Good? There's more than that, but those two are good. Everybody good? Okay, so, all right, so continuous, we got fenestrated, we got sinusoid. Okay, now we're going to go back to veins. All right, so now as we start moving up with veins, please note before we go any further that veins almost invariably have a wider lumen than arteries, right? Okay, and you saw that in the histology slide as well. Veins, of course, need less elasticity than arteries because the pressure is going to be way lower. And the pressure is going to be way lower, again, for multiple reasons. First off, they're further away from the heart, yes? Further away from the pump. Second, just the fact that you have a wide lumen will drop the pressure. Remember also that you had some fluid volume loss as you went through the capillary. All of those add up. And then one more thing. You've also got about twice as many veins as you do arteries. So if I sent all of this blood through 10 vessels and send it back through 20, isn't it going to be lower pressure? Yes? Okay. So you got way more veins than you do arteries. Your veins contain a disproportionate amount of your blood when you're at rest. So if you're just like sitting around not doing much, about 65% of your blood is in your veins at any given time. However, if you get up and start working out, what do you do to it? You shunt it really quickly back to the heart, right? And then you send it disproportionately to the arteries, right? Okay? And then if you settle down again, you're like, it's fine. Like the blood is not just sitting in your veins. What it's doing is it's not hurrying on its way back home, right? But if you decide you really, really need that blood, then it hurries on its way back home and then you send it to the arteries, right? So they call your veins a blood reservoir. They're kind of like holding on to it whenever you're really not needing it as much any other place, but it's not just pooling there because you know that would be bad. Okay, so So the veins contain up to 65% of your blood volume. Okay, so veins have a pressure issue, and the pressure issue can become problematic. So fluids, including blood, all want to flow from high pressure to low pressure. Like the reason when you turn on your faucet that stuff comes out is because it's flowing from high pressure to low pressure, and then gravity is also helping it, right? Okay, but if you use a hose, right, and you're holding it up like this, right, and you turn it on, if it's got enough pressure, it's still going to squirt out, right? Because high pressure or low pressure, right? So with veins, we run into some problems because the pressure is relatively low, and then if we add gravity against it, we can have some pressure issues. For instance, the blood in my legs is trying to run which direction through veins? Up, right? And in my arms as well. Also in my abdominopelvic cavity, it's trying to move up. Not particularly problematic in my head because it's trying to go down, right? But in my arms and my legs it is. So if you have a combination of low pressure and gravity, the blood will try to go backwards. And you definitely don't want it to go backwards. You want unidirectional flow of blood for efficiency of gas exchange. So what do you do about that? Well, there's two things. The first one is called the respiratory pump, and it works in combination with the valves that you have in veins. So let's talk about that one. Okay, so I have valves in some of my veins, not all of them, okay? I dependably have valves in the veins of my arms and my legs, and here is how I keep the blood from going backwards. Whenever my skeletal muscle contracts, right, it squishes on the vein, and what it does is it pushes the blood up because I've got a valve that won't let it go backwards. Those valves are one-way valves, right? You push it up, tries to go backwards, and they look like kind of like semilunar valves, right? If the blood gets caught in it, it's going to close the valve, so they push it up, and then the blood can't go backwards. That means that the more active you are, right, the better, they call it venous return, the blood returning through the veins to the heart. So basically what's happening is when you are active, you are actually squishing blood up, and it's going as it's making it up. Does that make sense? All the way up. So it's super important in your arms and your legs because, of course, you've got gravity really working against you in your arms and your legs. Does that make sense? Okay. So that's what the valves are about, and that's why I have them in veins and not generally in arteries. I don't really need them because the pressure is enough that the blood is unidirectionally flowing. Now, the other thing is that there's one other way that we actually keep our blood flowing through veins really dependably, and that is, okay, so it seems like you would need valves in the veins of the ventral body cavity, but you actually don't have any. You don't have any valves in the ventral body cavity at all. Even though the blood through, for instance, your inferior vena cava is definitely moving up, right? And it's wide, so it seems like, oh, it's wide, low pressure. You should need a valve in that. Huh?
Is it because everything's collecting there?
No, it's actually for a different reason. It's kind of counterintuitive, but it's also kind of interesting. So why do you not have any valves in your entire abdominopelvic cavity veins? It's because you breathe very readily. You breathe very frequently. So here's how it works when you breathe. I don't know if you've learned about the anatomy and physiology of breathing yet, but here's just a quick and dirty introduction to it. What happens before I inhale is my diaphragm is actually relaxed and bell-shaped, okay? Then when I inhale, my diaphragm pushes down. What does that do to the pressure in this cavity? Increases it. What does it do comparatively to the pressure in this cavity? Decreases it. So every time I breathe in, it actually squirts blood up the inferior vena cava, right? Breathe in, squirt. Breathe in, squirt. How frequently do you breathe? All the time if you're doing it, right? Right? So your breathing, your respiratory rate is so dependable that you actually don't need any valves in the entire cavity that you have right here, which is kind of cool, really, right? Okay, so that is called the respiratory pump. This is called the skeletal muscle pump. And collectively, what they are all trying to do is to improve blood flow through veins. That make sense? Okay. Okay, so now let's talk about those valves again for a second. So valves, let's go back to them. All right, so what if I had a weak valve? What if the muscle or the connective tissue in the wall of the valve was weak, and every time the blood sort of tried to go backwards right there and pooled around the valve, it sort of stretched the tissue? And that can happen because you might have a genetic predisposition for weak connective tissue or musculature in the wall of your valve. It also might happen because if I am standing, which means I've got gravity sort of working against blood flow, right? And I'm not wiggling around, then I've got sort of a double whammy with like the venous. And if I'm walking around, I'm totally cool because my skeletal muscle is contracting to help with the whole skeletal muscle pump. But what if I have a profession, for instance, in which I stand an awful lot? So if you have a profession in which you stand an awful lot, you're increasing the likelihood of ballooning out those valves over time. Just so much sort of pressure, like trying to go backwards. What? And it leads to varicose veins. So these are called varicosities when they balloon out. So here's some
Is there any way you can show?
nasty varicosities for you. So there's some nasty varicosities. What you have is ballooning out vessels. And the superficial ones are right under your skin, and they're going to show. Go ahead.
When my mom was pregnant, we gave it to her.
So you can better buy her Mother's Day present, for goodness sakes, right? She didn't even know she was going to have twins. Did you guys know that? Well, isn't that true, right? She didn't even know she was going to have twins. She just like had a baby, and then apparently wasn't done. And then which one of you came after?
I did.
You did. And then she was like, oh my gosh, guess what? I know you just had a baby. You know what would be awesome? Another baby. Said no one ever. Go ahead. Although I'm sure she's delighted. Go
Is
ahead.
there any way you can like shrink those valves?
You can't really shrink them. What you can do is change your activity patterns before you get them. Now I have a bit of bad news for you. There are certain professions that predispose you to varicose veins just because of the habits. And one of them is nursing. Yeah, nursing predisposes you to varicose veins because there is a lot of standing around during nursing. So what do? You be a wiggly nurse and not a still nurse, right? You know that the still nurses just sit around and get fat, right? Have you seen it? I know. Let's just be honest here for a minute. It's just us, right? So that profession has a predisposition to both obesity and varicosity, but it doesn't have to, right? You can get up and do stuff on a regular basis, right? Because you don't want to be the fat lumpy nurse. You don't, do you? Right? You don't. You want to be the really perky hot nurse, right? That's what you want to be. So varicosities occur quite often. Now, is it gender specific? No, it's not really. Women seem to have a slight predisposition to it, probably because of hormones, but they don't really know. But men get them too, but their legs are hairy, so nobody cares or notices. So you could just grow out your leg hair, right? That's a solution. It's a very practical solution. And then everybody wouldn't care. There's also a genetic predisposition, and the genetic predisposition, look at your parents' legs, specifically your mom's legs, and see if that happens. And then pregnancy increases the likelihood of all varicosities, okay? And heavy pregnancies, like with twins, will increase it even more. And why is that? Because you have so much pressure in your abdominopelvic cavity that it makes it harder to get the blood back up because blood flows from high pressure to low pressure, right? So pregnancy increases the likelihood of varicosities. It also increases the likelihood of the most socially inappropriate varicosities, which are called hemorrhoids. So hemorrhoids are when you have a varicosity that occurs in your anal canal and it balloons out. And I hate to tell you, but we're going to try to do something for birth control here. Pregnancy causes hemorrhoids, okay? Nobody wants to tell you that because they want you to get knocked up. But pregnancy changes everything, and it doesn't go backwards, just so you know, okay? I didn't teach you enough when you guys were like, took your maturation class, but you'll get it now, don't worry. So pregnancy causes hemorrhoids. Also, a profession in which you're sitting for hours and hours and hours and hours and hours and hours increases the likelihood of hemorrhoids as well. Like truck drivers get hemorrhoids, right? So that also does it. So that's the relationship between those kinds of things. Okay, so a vessel that is really, really wide, I'm not talking about like anything wrong with it, but remember that a vessel that is really wide is called a sinus. Can you see now why you have more venous sinuses than you do arterial sinuses? Veins tend to have a larger lumen to begin with. Okay, last little tidbit before you guys leave is to just get you started on the roots. We're not going to do anything hard with this. I want to talk about circulatory routes. So we are going to do pulmonary circulation, right? And then we're going to do systemic circulation, but one of those is a much longer story than the other, right? And which one is it? Systemic. So all I want to address right now is with pulmonary circulation, I want to remind you and put this together with the last set of notes and ask you, pulmonary circulation goes from which chamber through which organ back to which chamber? right ventricle through the lungs back to left atrium. Now I want to address something and see if you guys can conceptualize it. So I'm going to ask you two different questions about the same thing. First off, average at rest systemic circulation is 120 over 80-ish. 120 being the highest millimeters of mercury you get in an artery and 80 being the lowest. This is at rest, okay? So that's your variation that you see in there. But pulmonary circulation tends to be about 25 millimeters of mercury at the highest over 10 at the lowest. Okay, so two questions for you, and they're totally different questions. One of them, what anatomical difference accounts for the difference between the pressures in the two circuits? No, that's why it's okay. Now I'm asking you what generates the pressure difference. What is the responsible for generating the pressure difference in the pulmonary versus the systemic circuit? You can do this. No, I'm not asking why it's okay. I'm asking why it exists. What is the anatomical difference that generates the pressure differences between the two sides? You can do it. I know you can. There we go. Right ventricle versus left ventricle thickness. Remember that? Yeah, the left ventricle is a lot thicker and generates a lot more pressure, right? And now I'm going to ask you the question that you were expecting, and that is, why is that okay? Why is it okay that the pulmonary circuit is so much lower pressure than the systemic circuit? Because it's right there, and also there's not very many vessels, right? You don't have very many vessels to pump through. Does that make sense to you conceptually? Okay, we will stop there and we'll do the rest of the circuit on Monday after your quiz on the heart.